Our last lecture for this week is about bone and the histology of bone. Now, some general functions of bone is that it provides shape and support, a hinge for location and movement, houses the bone marrow, and stores minerals like calcium and phosphorus, is highly vascular compared to cartilage, and therefore can repair itself relatively quickly and is constantly changing. Bone tissue is made up of an extracellular matrix and cells. The extracellular matrix contains hydroxyapatite, which has calcium and phosphorus, as well as osteoid, which is type 1 collagen plus ground substance. And it has four major cell types: osteoprogenitor cells, or an osteogenic cell, osteoblast cells, osteocytes, and osteoclasts. Now, bone is remodeled so that body has access to calcium, the nutrient stored within, and triggers for remodeling could be mechanical forces, the parathyroid hormone, which activates osteoclasts, and calcitonin, which inhibits osteoclasts. Our osteoprogenitor cells are flat-shaped stem cells. They are derived from mesenchyme. They're located in the endosteum and the osteogenic layer of the periosteum, the outer surface of the bone, and they can proliferate and differentiate into osteoblasts. Osteoblasts, on the other hand, proliferate and secrete osteoid, which is type 1 collagen, and then they start the extracellular matrix mineralization by secreting alkaline phosphatase. They then get trapped in osteoid and become osteocytes, and they can also transform into inactive bone lining cells in the peri or endosteum. The osteocytes, as seen here, is kind of these black, almost oval shaped things with that look like they have projections, are the active cells that are enclosed in the bone matrix. They communicate with each other with these little caniculi through gap junctions in their caniculi, and they provide nutrient transport. They also are mechanosensory. They can sense small fractures or cracks in the bone. They have cytoplasmic extensions, as you can see in the caniculus, and they are located in lacuna. Osteoclasts are formed by fusion of blood monocytes. They are much larger and multinucleated, and they are eosinophilic in nature, meaning that they can be like a macrophage. They reabsorb bone and mineralize cartilage. So, looking at a cartoon version of them, they can release hydrolytic enzymes to digest the bony matrix. Their ring of adhesions can seal the reabsorption bay, and it releases hydrochloric acid, which is a really low pH that helps to decalcify the matrix beneath. When we look at immature bovine bone, the collagen fibers are kind of arranged every which way. They're very poorly arranged and organized. Therefore, this is a weak tissue mechanically. We find this in fetal tissues and also it arises in bone repair, but it's quickly remodeled. Mature lamellar bone, we can really see this is kind of immature woven bone, then gets remodeled into this picture where our collagen fibers are assembled into these parallel sheets, lamellae, and this is very mechanically strong, and it can be either classified as compact, as seen here, or as spongy, as seen here. Our spongy bone would be considered cancellous or trabecular bone. That's another word for it, versus compact bone is cortical or lamellar bone. This bone is constantly remodeled and it contains these osteons here, which are also called Haversian systems. The mode of bone lamellae are concentric rings or concentric layers, almost like a tree, and they surround this Haversian canal that you can see here. This is going to run lengthwise throughout the cortex of the long bone. Volkmann's canal, on the other hand, are connecting osteons running perpendicular to the Haversian canal, and it connects to the periosteum. Reminding ourselves of bone anatomy, we have articular cartilage on either end. This is of a long bone. We have spongy bone. We have our epiphyseal line, and this is the proximal epiphysis versus the distal epiphysis. We then have the metapiphysis, which contains the line, red bone marrow, and we have the metapiphysis here. We then have the diaphysis here, the kind of length of the long bone that has compact bone on the outside and endosteum, as well as then periosteum. And we have a medullary cavity with yellow bone marrow here and a nutrient artery. So what is periosteum? Well, it covers the outer surface of the bone and it's a fibrous, has a fibrous layer that's outer layer that's dense, irregular connective tissue. And it has lots and lots of blood vessels. It then has an inner osteogenic layer that has stem cells, osteoblasts, and osteoclasts. The endosteum covers the inner surfaces of the bone, and it's a single layer of osteoprogenitor cells. When we form a long bone, this is looking at a growth plate, we can think about the process of endochondral ossification, which occurs with a cartilage template. Examples are our length of our long bones, our vertebra, our pelvis, and even the base of our skull. So, what is this endochondral ossification? It's using a hyaline cartilage template surrounded by perichondrium, except for at articular surfaces, to then create an adult mature bone. The perichondrium of the diaphysis becomes periosteum, and a bone collar is formed which impedes nutrient diffusion out to the cartilage. Chondrocytes become hypertrophic and enlarge, and then they mineralize to calcify their matrix and undergo apoptosis, programmed cell death. Angiogenic factors that promote blood vessel development are released by these chondrocytes, and this is due to local hypoxia, and it recruits blood vessels to the area. These blood vessel buds burst through the bone collar and they deliver osteoprogenitor cells, osteoclasts, and hematopoietic stem cells. The osteoclasts start removing calcified cartilage matrix, and when osteoprogenitor cells come in contact with the remaining calcified cartilage, they transform into osteoblasts and start laying down bone. The primary center of ossification is formed in the diaphysis, kind of in the center. They then extend the bone collar, and there's cartilage at both ends of the diaphysis continues to calcify. The osteoclasts continue to remove that calcified cartilage, and osteoblasts continue to release osteoid, and the woven bone is formed and bone remodeling starts. The bone collar extends, and secondary ossification occurs in both or either epiphysis. So there's two regions of cartilage that do not calcify, and that's the epiphyseal growth plate and articular cartilage. So when long bones grow then, when we are achieving maximal growth, we have proliferation of chondrocytes within the epiphyseal plate. This is showing the growth plate zone. Once that stops, once we've achieved our maximum growth, that process will stop. All initial cartilage will be ossified except for the articular surface, and the epiphyseal plate will leave a remnant in adult long bones, which is called the epiphyseal line. This is made of compact bone between the epiphysis and the diaphysis. So looking at this growth plate zone, we see that this is the zone of ossification here in the metaphysis. This is showing our primary spongiosa and secondary spongiosa. This is showing our calcified matrix and cell death. This is showing the maturation of hypertrophy of our cartilage cells, the proliferative zone of our cartilage cells, and the reserve zone where matrix production is occurring. Now we can remodel long bones and change them as well, and remodeling is required as bones re-elongate, and it's a balance of preferential reabsorption of bone and deposition of bone. And we can see here when a bone grows, we had to resorb a portion over here, so we could add a portion up here. When fractures occur in adults, it looks much like fetal skeletal tissue development in that when we have a fracture, a break here, we form a cartilaginous callus, a soft callus, and then we replace it by a bony or a hard callus, and then it becomes mature normal bone again. Cartilage repair is challenging because it's avascular tissue in nature. We can see this break here, and chondrocytes are not really super mobile. There's also limited abilities of mature chondrocytes to divide and proliferate, and hyaline cartilage is prone to calcification and replacement by bone. Our last bone component is flat bone formation. This undergoes intramembranous ossification, where it's not using a cartilage template. And we have some examples, like the skull is one of the examples of what uses that. So in intramembranous ossification, we don't have a cartilage template. So instead, our mesenchymal cells aggregate where bone will form. They then differentiate into osteoprogenitors and blast cells, which secrete osteoid, which is non-mineralized. They then become separated but remain slightly attached by their cytoplasmic processes. It's really thin. The osteoid becomes mineralized. They then become trapped and become osteocytes. And then the tissue becomes vascularized. It brings in osteoclasts to remodel it. Here's some great self-study. I encourage you to cover up all the areas of color and fill in the blanks to study this content and material. Same here, talking about bone remodeling and then bone formation. And that's all for this week for histology.